Meiosis in humans is the division process that takes us from a diploid cell, one with two sets of chromosomes, to haploid cells, ones with a single set of chromosomes. In humans, the haploid cells made in meiosis are sperm and eggs. When a sperm and egg join in fertilization, the two haploid sets of chromosomes form a complete diploid set, a new genome.
After crossing over, the spindle begins to capture chromosomes and move them towards the center of the cell, metaphase plate. Each chromosome attached to microtubules from just one pole of the spindle, and the two homologous of a pair bind to microtubules from opposite poles. So during metaphase 1, homologous pairs, not individually chromosomes, line up at the metaphase plate for separation. When the homologous pairs line up the metaphase plate, the orientation of each pair is random. In anaphase 1, the homologous are pulled apart and move apart to opposite ends of the cell. The sister chromatids of each chromosome, however, remain attached to one another and don't come apart.
During telophase 1, the chromosomes are enclosed in nuclei. The cell now undergoes a process called cytokinesis. Divides the cytoplasm of the original cell into two daughter cells. Each daughter cell is haploid and has only one set of chromosomes, or half of the total number of chromosomes of the original cell. Prophase 2. During prophase 2, chromosomes condense and the nuclear envelope breaks down, if needed. The centrosomes move apart, the spindle forms between them, and the spindle microtubules begin to capture chromosomes. During metaphase 2, the centromeres of paired chromatids align along the equatorial plate in both cells.
Then, in anaphase 2, the chromosomes separate at centromeres. The spindle fibers pull the separated chromosomes toward each pole of the cell. Finally, during telophase 2, the chromosomes are enclosed in the nuclear membranes. Cytokinesis follows, dividing the cytoplasm of the two cells at the conclusion of meiosis. There are 4 haploid daughter cells that go on to develop into either sperm or egg cell.
Okay guys, let's talk about mitosis. Mitosis is the phase of the eukaryotic cell cycle that occurs between DNA replication and the formation of two daughter cells. Mitosis has 6 substages: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. First, interphase. During interphase, the cell grows and the nuclear DNA is duplicated. Interphase is followed by mitotic phase. During the mitotic phase, the duplication chromosomes are segregated and distributed into daughter nuclei. The cytoplasm is actually divided as well, resulting into 2 daughter cells. First, and the longest phase of mitosis is prophase. During prophase, chromatin condenses into chromosomes, and the nuclear envelope, the membrane surrounding the nucleus, break down.
In metaphase, the spindle reaches maturity and the chromosomes align at the metaphase plate, a plane that is equally distant from the two spindle poles. During this phase, a number of changes occur. The nuclear membrane disappears completely. Polar fibers, microtubules that make up the spindle fibers, continue to extend from the poles to the center of the cell. Chromosomes move randomly until they attach at the kinetochores. to polar fibers from both sides of their centromeres. Chromosomes align at the metaphase plate at right angles at the spindle poles. Chromosomes are held at the metaphase plate by the equal forces of the polar fibers pushing on the centromeres of the chromosomes. In anaphase, the paired chromosomes, sister chromatids, separate and begin moving to opposite ends of the cell. Spindle fibers not connected to chromatids lengthen and elongate the cell. At the end of anaphase, each pole contains a complete compilation of chromosomes. During anaphase, the following changes occur. The paired centromeres in each distinct chromosome begins to move apart. Once the paired sister chromatids separate from one another, each is considered a full chromosome. They are referred to as daughter chromosomes. Through the spinal apparatus, the daughter of chromosomes move to the poles at the opposite of the cell. The daughter chromosomes migrate centromere first and the canticore fibers become shorter at the chromosomes near a pole. In preparation for telophase, the two cell poles also move farther apart during the course of anaphase. At the end of anaphase, each pole contains complete compilation of chromosomes.
Telophase. Telophase is the fifth and final phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Telophase begins once the replicated paired chromosomes have been separated and pulled to opposite sides or poles of the cell. During telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. The chromosomes begin to uncoil, which make them diffuse and less compact. Along with the telophase, the cell undergoes a process called cytokinesis that divides the cytoplasm of the parent cell into two daughter cells. Telophase. Telophase is the fifth and final phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Telophase begins once the replicated paired chromosomes have been separated and pulled to opposite sides or poles of the cell. During telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. The chromosomes begin to uncoil, which make them diffuse and less compact. Along with the telophase, the cell undergoes a process called cytokinesis that divides the cytoplasm of the parent cell into two daughter cells.